All right, I'm going to summarize 2.1, 2.2, and 2.3 real quick using this study sheet that you guys can use. So 2.1 is understanding how living systems are organized in that hierarchy of structural levels that are all interacting with each other. So we're going to real quickly look through our structures and as well as their interactions. 2.2 talks about how each subcellular component is contributing to the function of the cell as a whole, what the structural features are that allow that component to capture, store, and use energy, and how does the surface area to volume ratio affect the exchange of materials between cells or organization in the environment. That's your 2.3. So quick summary, your nucleus, this is your center of operations. The structure of your nucleus, it's located near the center. It's enclosed in nuclear envelope. It has that double membrane. The function of our nucleus is to store DNA, synthesize RNA in the nucleolus. Now, your ribosomes are made of ribosomal RNA. So that's your rRNA and protein. So as you can see right here, your nucleolus is creating those ribosomes and sending them out. Now, once those ribosomes are sent out, over here, we see that they can either be free floating or attached to the endoplasmic reticulum. And we looked at that yesterday, that the ones that are free floating in your cytoplasm, those are the ones that are going to be used for that specific cell, and then the ones that need to be packaged out are the ones that are making the proteins that are going to be packaged out are the ones that are attached onto the rough endoplasmic reticulum. Okay, so our rough ER, this is our manufacturing tunnel system. Okay, so the structure on this rough ER is we have our network of membranes. It's forming the network of tubules and sacs. Okay, and our function is to transport material within the cell. You have two components on the ER. We have our smooth ER and our rough ER. Your smooth ER is carrying out metabolic processes such as making lipids, making phospholipids, cells. Your rough ER, this is that manufacturing component. Okay, so your rough ER is going to modify proteins that the attached ribosomes have synthesized if needed, and then prepare them for their function. So as you see right here, we have our protein made by our ribosome, and your endoplasmic reticulum is taking that protein, putting it into a vesicle, and sending it out to the Golgi body. Now, within the Golgi body, this is our processing and sorting. The Golgi body, they're going to modify those proteins according to whatever they're going to need to do. So you see right here that it's modifying that protein, shaping it, attaching either a lipid or a carbohydrate depending on what is needed. Okay, so once it modifies and sorts them, it ships them out to the rest of the cell in vesicles, and that process is called your exocytosis. And those vesicles are going to look just like the vesicles they were brought to the Golgi body in by your endoplasmic reticulum. All right, plant cells. In plant cells, there are some unique components that animal cells will not have. So for our ER, your plant cells have an additional function, and that's to play the central role in cell-to-cell communication in our specialized cells. They're also going to store proteins and have additional enzymes and proteins that are needed for plants to make and store lipids. On the Golgi complex, within plant cells, the Golgi complex is the biosynthesis organelle, which is manufacturing large amounts of polysaccharides that plants are going to need to build their cell walls. Now, Golgi complex is also in charge of creating lysosomes. So these are your recycling bins, the cellular recycling bins. Their structure, they are membrane enclosed sacs of hydrolytic enzymes. These are your digestive. Their function is to digest all major types of macromolecules as well as any worn out materials that may be floating around the cell. The process that our lysosomes are using to degrade old organelles is known as autophagy. Now, lysosomes are rarely found in plant cells because in plant cells, the central vacuole already performs that function. Now, how do the ER and Golgi body contribute to the function of the cell? This is your 2.2. Your rough ER is providing mechanical support. So look at what it's doing right here. And it's also creating or carrying out protein synthesis, which is going to be helping the rest of the cell. And it's helping with intracellular transport with those vesicles. Your Golgi body is helping your protein, it's modifying and packaging and sorting and sending those proteins out. And your lysosomes are helping because they are recycling the cell's organic material and carrying out apoptosis, which is programmed cell death. All right, vesicles and vacuoles. Vesicles are mainly your transport, vacuoles are mainly your storage. Now, vesicles, they are small compartments that are enclosed in a lipid bilayer membrane filled with fluid from the parent organelle. So, for example, if it was the Golgi, it's filled with fluid from the Golgi. If it's the one that's leaving endoplasmic reticulum, it's filled with fluid from the endoplasmic reticulum. The function of your vesicles is to transport those molecules from one organelle to the next. So think about when they went from the rough ER to the Golgi. This is the transportation system that's linking all of our organelles together. There is a special type of vesicles called peroxisomes. This type of vesicles comes from the ER or other peroxisomes. Okay, so they are structurally similar to lysosomes, but they are bigger in size than lysosomes. And we actually find these only near the mitochondria and chloroplast specifically. And their function is to detoxify, break down toxic substances that may be somewhere. Now, one of the key differences between your lysosomes and your peroxisomes. Lysosomes are more about keeping everything at the right level. So there are certain substances that may only be harmful to the cell if we have it in an excess amount. So lysosomes will go in and they will maintain the right amount by killing anything that becomes extra. Peroxisomes, however, these are the ones that are going in and they're killing anything that may be toxic. So for example, ethanol is going to be toxic. So the peroxisomes will go in and they'll break that ethanol down into water molecules and oxygen. Vacuoles, these are your storage. Their structure is large membrane enclosed fluid-filled sacs. Function is mostly storage. Now, we can have different types of them. We can have contractile vacuoles. We can have food vacuoles. Now, in plant cells, your central vacuoles are larger, and their function includes maintaining water concentration and maintaining the cell's shape as it enlarges. Your vacuoles are important and their important function towards contributing to the cell as a whole is to store and release macromolecules and cellular waste products. Now, vesicles compared to vacuoles. Now, both of them have the same function. They're both somewhat storage and transport. However, their structure is going to be somewhat different. They do both have membrane-bound structures, but vacuoles are going to be larger. And vesicles, their membranes are made in a way that allows vesicles to fuse with other membrane systems within the cell. So, for example, when the vesicles left the Golgi body, they went and they fused. When the vesicles left the rough ER, they went and fused into the Golgi body. And the same thing when the vesicles leave the Golgi body, wherever they're going, they'll fuse over there. Now, organelles that are not fully dependent on the nucleus. This is our next category of our organelles. We have our mitochondria and our chloroplasts. Okay, so we've talked about these before. And our last component, our last category is keeping it all together. Okay, so your cytoplasm is the component that has everything contained in the cell except for the nucleus. Structure and function, you've got your cytosol within the cytoplasm. That's your fluid part of the cytoplasm. It's containing the water, salts, and any other chemicals that are there, and it has a jelly-like consistency. Your cytoskeleton, this is the structure, which is our network of fibers that stretch throughout the cytoplasm. Its function is to provide that intricate framework that we need for support and movement within the cell. Now, cytoskeletons associated in specialized proteins that are involved in movement such as muscle contractions. We have our plasma membrane, which is the structure that surrounds the cell. It is selectively permeable, meaning it will let certain substances in, and it has certain substances it will not let in. The function of your plasma membrane is to control transportation into and out of the cell. Your cell membrane is made of proteins and fats, and this is where one cell ends and the other cell begins. Now, 2.3. How does surface area to volume ratio affect the exchange of materials between our cells, organisms, and the environment? Your ratio affects the ability of your biological systems to get the resources that they need, get rid of their waste products, as well as exchange the chemicals and energy with the environment. So, for example, mitochondria needs to get those macromolecules and convert them into ATP, which the cell can then use. So that interaction is dependent to that surface area to volume ratio. We need a larger surface area to volume ratio if we want to be able to adequately exchange our materials. So that means that our cell size and shape is restricted. We need a larger surface area to volume ratio, which means we need to have smaller cells. We have smaller cells, we can more efficiently exchange those materials with the environment. If we were to increase the volume of our cell, we end up decreasing that ratio. And if we decrease that ratio, there's less exchange of materials with the environment. And if they're not getting that material from the environment, the demand within that cell for internal resources goes up. The more complexity we have in our cellular structure, meaning folds, the better exchanges we're going to have of materials with the environment. So, for example, mitochondria, the more folds you have, the more ATP you can produce. Chloroplast, the more stacks you have, the better it can do photosynthesis. Now, the larger an organism, the smaller their surface area to volume ratio. So this would then affect properties like the rate of heat exchange with the environment. So remember, the larger the ratio, the better the exchange. So you don't want to get confused with those terms. When we're saying larger ratio, that's a larger ratio, which would mean that the substance itself is actually smaller. Alright, so I'm going to send this study sheet out to you guys. Now, I know that on here, these are able to play on my